So I've had the great fortune of working with Andrew for well over a decade. Andrew is an NHMRC Principal Research Fellow and Associate Director at the Florey Institute of Neuroscience and Mental Health, where he's the head of the Division of Behavioural Neuroscience and runs the Addiction Neuroscience Laboratory. Andrew's published over 200 original articles and reviews, is highly cited and internationally respected, and was recently President of the Asian Pacific Society for Neurochemistry, Senior Editor of the British Journal of Pharmacology, and is currently Deputy Editor-in-Chief of Pharmacology Research and Perspectives, Associate Editor of both Neurochemical Research and the Journal of Pharmacological Sciences. And Andrew will present today on stress and relapse, insights from neuroscience.
Okay. Thanks, Dan, and thanks for the invitation, and I hope you enjoy my presentation. So I'm going to start with a quote from Edgar Allan Poe. I have absolutely no pleasure in the stimulants in which I sometimes so madly indulge. It has not been in the pursuit of pleasure that I've perilled life and reputation and reason. It has been the desperate attempt to escape from torturing memories, from a sense of insupportable loneliness and a dread of some strange impending doom. Just think about that. Last year, there was a stress and wellbeing survey in Australia. 35% of Australians surveyed reported significant distress. 61% of those that reported severe distress reported that they drank alcohol. 57% reporting severe depression reported that they drank alcohol. And 66% reporting severe anxiety drank alcohol. So that's a lot of people drinking alcohol as a consequence of some form of distress. A number of years ago, Solomon and Corbett put forward what's called an opponent process theory. It's a psychological theory that may, psychologically at least, explain the transition to addiction. So if we look over on the left-hand side, if we think of this as a hedonic scale, and plus 100 in state A is euphoria, and plus 100 in state B is dysphoria, they argued that in early drug use, the acute euphoric effect of the drug far outweighed the off phase of the drug, the dysphoric effect when you're coming down off the drug. And they also argued that with ongoing sustained drug use, that this almost mirrored so that there was less of an acute euphoric effect to taking the drug due to tolerance and a range of other factors, but an increase in this dysphoric component acutely when coming down off the drug. This was a psychological argument. Koob and other colleagues in the States put a neurobiological framework on this and called it allostasis, with this notion of further reduction of this homeostatic set point. And that was based on behavioural observations and based on empirical observations of dysregulation of transmitter and peptide systems in key brain regions, that some of which Dan was mentioning a moment ago. If we think about relapse, though, the title of my talk and the content of my talk today is about relapse. People relapse for different reasons. It may be a cue has precipitated the relapse. It may be a priming of a drug, for example, passive inhalation of smoke, for example, or it may be an acute stressor. Now, irrespective of these precipitants of the relapse, there's a common base network in the brain that drives that behaviour. However, there are also specific subcircuits that are implicated in different forms of relapse. So, for example, if you're relapsing from a cue, the basolateral amygdala is a critical component in the circuitry that drives that behaviour. Whereas if it's a stress, the central amygdala and the bed nucleus of the stria terminalis are critically implicated in that behaviour. So if we're thinking about stress, because stress is the content of today's talk, circa 2016, a recent review came out saying this is the circuit and the chemistry behind stress-induced relapse to drug seeking, based on predominantly animal models, but also on human experimentation as well. And I'll draw your attention to these two cell groups in blue that I mentioned to you a moment ago that are critical for stress-induced relapse, the central amygdala and the bed nucleus of the stria terminalis. And the prototypic stress neuropeptide corticotrophin releasing factor, CRF or CRH, has been implicated in that behaviour, acting outside of the hypothalamus in these two structures. As a consequence of a lot of that research, there was great effort put in pharma and in academic laboratories, particularly in the USA, to develop small molecule antagonists that would block this stress-induced CRF system. And so a whole range of chemicals, one related here called MTIP, is a selective CRF1 receptor antagonist. It was developed, it was characterised, and it was shown in animals to very efficaciously reduce withdrawal-induced anxiety following chronic alcohol binging. And also it was shown here in a model of relapse that it was effective at dramatically reducing stress-induced relapse behaviour in animals that had been rendered dependent upon alcohol, but not in animals that were not dependent on alcohol. Okay? And they found similar findings in genetically engineered animals, or specifically bred animals that were alcohol preferring, as they found in outbred dependent animals, should I say. So far so good. Move forward about 8 or 9 years, clinical trials on selective CRF1 receptor antagonists in either male or female cohorts, predominantly again throughout the USA, didn't translate. No translation at all. In fact, there's a quote out of one of these papers: the findings do not support a clinical efficacy of CRF1 receptor blockade in stress-induced alcohol craving and relapse. So, if we go back to this proposed circuit and the chemistry behind this circuit for stress-induced relapse, that clearly raises a few questions. If we know that these two structures here are critically required for this behaviour to occur, and that something that's unique to these neurons within this circuit is CRF, and it looks like it works in animals, but it doesn't translate in humans, does it mean the animal models are wrong, or does it mean that there's more to it, more complexity to it than is shown on this cartoon? So we can immediately ask a couple of questions based on this collective evidence. Is this the full picture? And secondly, or related to that question, we can make a second question, which is, can we modulate this stress relapse circuitry another way? So can we, for example, modulate the activity of the neurons in this subcircuit a different way, rather than actually going for CRF? Can we tone down that circuit a slightly different way that may have greater effect? Well, let's see. I'd like to introduce you now to a neuropeptide that was discovered at the Florey about 15 years ago. It's called Relaxin III. It's a member of this superfamily of relaxin and insulin-like neuropeptides, or peptides in general. Relaxin III, it's a misnomer really. The reason it's called Relaxin III is because it was the last peptide discovered in this family, despite the fact that it is in fact the ancestral peptide of this family. And the family were called relaxins because initially Relaxin 1 was shown to be critical for uterine relaxation during parturition. Needless to say, it's the ancestral peptide in this family, and it's highly conserved. It's found in rodents, in primates, in humans, and all the way back down the evolutionary tree to zebrafish. It acts upon this cognate receptor called RXFP3, which stands for relaxin family peptide receptor number 3. You'll notice that it can also act at some of these other receptors, but the key thing is that none of the other peptides in this family of neuropeptides has the ability to act upon this receptor. So Relaxin 3 is the only peptide in this family that can activate that receptor. Okay? Okay, so I told you it's a neuropeptide. Whereabouts is it found? It's predominantly found in a very small structure in the pons in the brainstem. It's at the level of the locus ceruleus, but it's directly in the midline. Here's a picture of some immunostaining showing neurons that express this neuropeptide in the nucleus incertus. There is also a scattering of these neurons found located just immediately ventral of the nucleus incertus in a structure called the pontine raphe. There's also an even smaller scattering of these neurons in the ventral parts of the periaqueductal gray, and an even smaller scattering in this region of the mesencephalon immediately dorsal to the substantia nigra. This is where the dopamine cell bodies are found. Okay. In addition to that, if you look at down through an electron microscope, you see these dark circles here. This is a terminal. This is a presynaptic terminal. This dark line here is the synapse, and this is the postsynaptic cell. And what you can see in these black circles here, these are vesicles that store neurotransmitter, and this is immunoreactivity for this neuropeptide in the presynaptic vesicles that store neurotransmitter ready to be passaged across this membrane and released to convey a message to the next cell. If we look at where this structure is hooked up in terms of the circuitry, this very small structure in the pons actually has this very profound ascending pattern of innervation of structures implicated in feeding. It's an orexigenic peptide. Structures implicated in stress reaction integration. Structures implicated in reward. And structures implicated in memory and decision making. So if you think about that, it's going to all the right places that we've just mentioned, that Dan mentioned in his introduction and that I showed you in that cartoon for the circuitry that underpins this behaviour. So to synthesise a whole lot of findings that have been made along this system, because I don't want to bore you with that, I want to kind of cut to the chase, we know that compounds that activate this receptor enhance feeding. It's orexigenic, so acutely and chronically. It will make animals eat more and they will put on more weight. Antagonists, however, do not reduce feeding. What they will do is they will block agonist-induced feeding, but the antagonist by itself will not turn off feeding. Okay? So that would suggest that while activating the receptor can promote feeding, there's not a tonic role of this system in maintaining feeding on a day-to-day basis. But if you think about it from a therapeutic perspective, it's actually quite a good thing, because if you want, for example, if we take alcohol, which has caloric value, if you want to reduce someone's alcohol consumption, you don't want to stop them eating at the same time. So from that simplistic perspective, it's a useful observation. So based on that rationale and a bunch of other findings that we had, we asked this question: does this system modulate alcohol intake, ingestion, consumption, and also does it actually modulate the seeking, the active seeking of alcohol, okay, which would precede the consumption? So I'm going to show you a video here, and it does work because I checked it a moment ago. Here's a rat. He's going to press this lever here. When he presses the lever 3 times, there's a little disc here, which is a cue light. That's going to illuminate for a short period of time. The illumination of that cue light is going to coincide with the delivery of 100 microlitres of 10% ethanol to a receptacle that's in this magazine. And what you'll see is the rat will perform the task. The light will come on, he'll go into here, he'll consume the alcohol. When the alcohol's been consumed, he'll go back and repeat the task. 3, light goes on, he drinks the alcohol. He's not just watching the light go on and off, he's not doing it just to pass the time of day. You see, in a 20-minute period, these guys will do this behaviour 100 times. 100 times. And they'll drink to intoxication. It's a really good model of a binge drink, okay? So if we use this paradigm, animals voluntarily self-administrate the alcohol. There's no coercion whatsoever. They do it voluntarily. So this is baseline responding, you see, 100 lever presses for alcohol. This is the alcohol lever, the black lever. The white lever is a second lever that gives them water. So you can see they've got just as much capacity to self-administer water as they have alcohol, but they clearly prefer the alcohol over the water. We inject them with vehicle, just an inert solution that the drug's dissolved in. It has no effect on that behaviour. They consume the alcohol in a similar manner to the baseline, and there's the similar discrimination between the lever that delivers the alcohol and the lever that delivers water. And if we inject these animals with this drug here, R3B1-22R, it's a kind of telephone number name, but all you need to know is that from a pharmacological perspective, this drug is a highly selective antagonist for that RXFP3 receptor. And you can see, we see this really nice dose-related reduction in alcohol self-administration when we treat the animals with this drug. And it has no impact on their propensity to consume water, it's only reducing their alcohol intake. Okay. We had another control experiment. If you see here, at 30 micrograms, it's just about wiped out the alcohol self-administration, and 10 is probably about a 50% reduction. We take those 2 highest doses, we train another group of animals to self-administer sucrose. No effect. Same line of animals, performing the same task. The only difference is these guys have a choice of sucrose versus water. These guys have a choice of ethanol versus water. So we see the same drug, same paradigm, same rats, has a specific effect on the reduction of the consumption of alcohol and has no effect whatsoever on the consumption of sucrose. So how do we go from there? Because that's just self-administration, that's consumption. How do we go from there to relapse? Well, we saw this phase. I showed you a video here when the animal had learnt the task and was self-administering. So what we can do here is we can effectively send them to rehab by changing the rules such that now, when they press that lever, there's no alcohol. And they'll reduce performing that behaviour because now there's no consequence. When they get to this point, we can then provide them with a particular stimulus to see if that will precipitate a relapse-like behaviour. And that could be presenting them with a cue, it could be an acute stressor, or it could be a drug prime. And when you do that, you see this profound reinstatement of that drug-seeking response. Okay, when we do that, this is relapse or reinstatement precipitated by a cue. When we pre-treat the animals with the drug, we attenuate it, doesn't block it, but again, you see the behaviour is directed towards the active lever that gave alcohol, not the water lever. If we do a stress-induced reinstatement, we see a profound reinstatement that is completely prevented by this drug, completely prevents a stress-induced relapse. If we do the same experiment for relapse to sucrose seeking, no effect. Okay, no effect. Okay, so we can ask a question, whereabouts in the brain is this system acting to modulate this behaviour? And there's a clue. The clue is that it was more effective on a stress-induced relapse compared to a cue-induced relapse. So if we go back to this circuit, we honed in on the bed nucleus of the stria terminalis. For this reason, here's a section of the bed nucleus showing the input from the nucleus incertus. So it receives a neural input from that structure in the brainstem that I told you where these neurons reside. And here's a picture in a rat brain of the receptor for this peptide on the neurons, and here's a reporter mouse, again showing the receptor protein on neurons in the bed nucleus. So this structure receives an input and it contains the receptors that the peptide acts on. So what we did here is we restricted our drug treatment solely to that structure. We injected the drug directly into the bed nucleus of the stria terminalis, and we see we got an attenuation, not a complete inhibition like we saw with the previous treatment that was getting the drug everywhere, but when we restrict the drug solely to the bed nucleus, we get a reasonably good attenuation, at least a 50% attenuation of that stress-induced relapse. And if we do control injections immediately adjacent to this structure, we have no effect. So there's an anatomic specificity. So now we can ask a question, well, how does stress regulate this Relaxin III system that could make it interface with the relapse circuitry? So I'll just take you through the last few slides. Okay, this is that immunolabelling I showed you of the relaxin peptide in the nucleus incertus back in the brainstem, and it just so happens that these neurons co-express the receptor for corticotrophin releasing factor, CRF1. What's more, if you inject animals with CRF, we get a profound activation of these relaxin 3 neurons in the brainstem, and this is showing electrophysiologically the activation of these neurons following an ICV or an intracerebral injection of CRF activates these neurons. Furthermore, if we apply an exogenous stressor like a restraint stress to the animal, what that does is it increases the expression of this peptide relaxin 3, and we can completely prevent that by pre-treating the animals with one of these selective CRF1 receptor antagonists. So, we know from that nucleus incertus relaxin 3 neurons are sensitive to both exogenous CRF and to exogenous stressors. So we can ask a very simple question. Does a CRF input to the nucleus incertus regulate stress-induced reinstatement of alcohol seeking? So here's the final couple of data slides. Over here on the right are sections through the brain, and you'll see the green symbols are where injections were placed directly inside this nucleus, and the red symbols are control injections immediately adjacent to see if there's an anatomically specific effect. And we see here that animals reinstate to a stressor, and when we inject a CRF1 receptor antagonist directly into this structure in the green dots, we get an attenuation of this behaviour. When we inject them around the edges of this structure, we have no effect. Okay, so it's an attenuation, it's not a complete inhibition, it just means that there's more than one locus where this effect is happening, which I've shown you other loci earlier on anyway, so that's no great surprise. That's the CRF1 antagonist. If we do the same experiment, again, green are the direct hits and red are the control injections with a CRF2 receptor antagonist, the other receptor for CRF, we see we get no effect. We get no ability to reduce this behaviour, either when we inject within the nucleus incertus or outside the nucleus incertus. So from a summary perspective, we believe now that what's happening is CRF is, well, should I say, stress activates the nucleus incertus that projects up to the bed nucleus of the stria terminalis and drives relapse by activating this RXFP3 receptor. And part of the circuit that is implicated in that is a source of CRF, at this point unknown, that activates a CRF receptor within the nucleus incertus to turn this system on. So therefore, we've got a very simple question that we're pursuing at the moment. Could this receptor, RXFP3, represent a more amenable target than CRF1? It could potentially have fewer on-target side effects just due to a more restricted distribution in the brain, if nothing else. So if we go back to this putative circuitry that I showed you early on, I'd like to add something to it. Excuse my scrawly drawing, but I'd like to suggest that this nucleus incertus in green here that innervates not only the bed nucleus of the stria terminalis, but I didn't have time to tell you today, we've done very similar experiments in the central amygdala and get very similar results. I'd like to suggest that this peptide input to these two structures should also be added to this circuitry. Obviously, is this still the full picture? Probably not. I'd just like to finish off by acknowledging the people that did the experiments, collaborators that assisted with reagents and various other things, and the people that paid for me to do these experiments. And I started with a quote, so I'll end with a quote. This is politically incorrect, but it's possibly my favourite quote of all time. In 1969, I gave up women and alcohol. It was the worst 20 minutes of my life. So this is George Best. Many of you in the room will recognise George Best. This was him playing soccer for Manchester United, within probably the top 2 or 3 people that have ever played football, soccer in the world. Trouble was he was an alcoholic. Here he is in hospital recovering from a liver transplant, and he came out of hospital, recovered from his liver transplant, started consuming alcohol again and died, unfortunately. But to me, that quote sums up his problem in a nutshell. Anyway, I'll stop there and take questions.
So questions from the audience? So just while you're thinking, I suppose many of the people in the audience will work in clinical services or work in those clinical settings. I suppose my question then is around what you're identifying is potentially a circuitry that sort of hasn't really been explored before.
Yeah, that's right.
Sort of what is it relationship? What do you think the relationship is between sort of people, obviously who become stressed to make them vulnerable to addiction? So what happens in this circuitry, and how far are we away from thinking about sort of potential therapeutic options clinically?
Sure. I mean, I think, thanks for that, Dan. I mean, the problem is, when you look at a diagram of a circuit for whatever it is, whether it's relapse, whether it's feeding, whether it's scratching your big toe, you see a 2-dimensional picture and you think that's it. But the reality is it's not it. That's all that's known at this point in time, and people don't know if anything else is part of that network because they've never chosen to look, or they've never had reason to look. Well, we had reason to look here, and we found something that clearly interfaces on this known circuitry. So it tells me that the circuitry needs to be redefined, and it also tells me that there's possibly other unknown factors that still feed into this circuit, so there are potentially other ways to modulate this. We know that these neurons in the brainstem are highly responsive both to acute stressors and to chronic stressors. We know that. We don't know that in humans, but we know that in mice, we know that in rats, and we know that in primates. So it seems to be conserved. We know that also in zebrafish. So we see changes in this structure just as a consequence of stress. And not only that, obviously we know that the amygdala, particularly the central amygdala, has profound changes as a consequence of long-term stress. And part of that could be due to increased input from this structure that hasn't ever been considered to be the case in the past. Because, as Dan said in his introduction about, the disease model and dogma, people tend to stick to their own dogma and don't like to necessarily open their eyes to other possibilities. And whereas we're, just going with our eyes wide open and seeing what we get and using a biological scientific approach to take that forwards.
Any other questions? Nobody's got another question. I'm going to ask another one. It's going to link, I'm going to ask Murat to come up in a second. I suppose one of the questions is, obviously you're focusing on sort of pharmacological targets for changing the relaxin system in terms of antagonists and agonists.
In this example, yes.
So obviously one of the things that we'll be talking about today is sort of non-pharmacological approaches.
Absolutely.
So things like mindfulness, exercise, yoga, etc. Is there any studies looking at how behavioural sort of interventions can sort of modulate the system?
Yeah, absolutely. Not the relaxing system per se, we haven't looked at that, but I mean, you're quite right. I'm not suggesting that relapse should be treated solely by chemistry, solely by pharmacology. I think there's multiple approaches, and I think behavioural approaches are critical as well. And one of the examples of that we've been actively pursuing is looking at that process of extinction. So if you remember that cartoon of when the animals were self-administering, and then I said we put them into rehab and they stopped their responding, and then we provoked a relapse type response. Well, that process is known as extinction. It's new inhibitory learning. And if we've got some good empirical evidence that, in fact, Christina, who's in the audience here, was part of those experiments. If we can consolidate and enhance that extinction learning in various ways by knowing the fact that this form of inhibitory learning is context dependent, so that if you want to extinguish the salience of a cue that's paired with a drug, it's got to be done in the right context, not in an inappropriate context. And we know that if we can actually develop behavioural paradigms that really consolidate and improve and enhance that process of inhibitory learning, it does give in itself, without any chemical treatment, greater protection against relapse than not doing that. So absolutely, it's always going to be a combination of both. So another way of looking at it is thinking of trying to identify cognitive enhancers, for example, for want of a better word, that will facilitate that process of extinction learning. Because that in itself, well, if you think about it, that's only a short-term drug treatment. It's not a chronic treatment every day. It's a treatment that's paired with that behavioural therapy to enhance and strengthen that behavioural therapy to increase the resistance and the reduce the susceptibility potentially to relapse going forward. So both approaches are critical.
Thanks, Andy. I think that's enough time to keep us on track. So please thank Andy again for a great talk.